Greetings. My name is Brandon Ogbunu. As of July 1st, today, I'm an assistant professor in the Department of Ecology and Evolutionary Biology at Yale University, and I'm really excited to be a part of the Genomic Forecasting Symposium. Now, I confess that I'm really bad at giving 15-minute seminars. I'm never on time. So what I'm going to do is give you more like a 7-minute seminar that I think will probably stretch a few minutes, and that way that'll give me an opportunity to make one point very, very clearly. The title of the seminar is Disentangling the Forces Underlying Adaptive Landscape by Environment Interactions. This is the obligatory historical slide where we kind of talk about someone from years past. And I think Darwin, of course, talked about it is interesting to contemplate a tangled bank. Darwin was referring to the interactions between species and that the forces of natural selection play out in this complex interaction. And this is often invoked in the ecological context. But I would argue that even cutting-edge problems in modern evolutionary genetics can be considered something similar, right? So we can kind of talk about this version of the central dogma, where you have all of this kind of genotypic information, right, that has to be that is translated in some capacity into and mapped onto a phenotypic space or a fitness space or what have you, right, which is also a tangled bank. And I would argue that the act of predicting anything, be it the weather or the outcome of a sporting event, and certainly kind of the complicated phenomenon that is genotype-phenotype mapping, requires that we understand how these pieces work and interact with each other such that we can map them from one side of the information, in this case, to the other. This is, of course, what machine learning is famous for being able to, and sometimes successful and sometimes not, and being able to kind of disentangle the information such that it can make reasonable predictions for what's going to happen, right? And more functionally, disentangling involves understanding this flow of information, right? And we can ask kind of specific questions, like what are the forces that complicate these relationships? There's a lot of things that are kind of in this map that are kind of tuning it in terms of how the genotypic space is mapped. What tools can we use to better understand it? And how can we frame it in order to get a better handle on it, right? Even more specifically than that, right, these kind of modern extended evolutionary synthesis concepts and really, really modern hippy-dippy evolutionary concepts like plasticity and evolvability and epigenetics and what have you are really are things that are firmly couched in kind of being able to understand the information flow here. Plasticity, of course, which we'll cover a little bit later, is about kind of performance across different performance in different environments, right? Today we'll focus on epistasis quite a bit. It'll be one of our folks guide for today. You might have seen this kind of consideration of the problem, which was very, very recent, which talks about kind of genotype and then having higher levels of phenotypes. This is kind of one way that people consider this problem. But I was really, really excited to see a different formulation of this problem, really, really humbled and really, really pleased from Dr. Oman and colleagues. This recent preprint, The Consequences of Single Locus and Tightly Linked Genomic Architectures for Evolutionary Responses to Environmental Change. And this paper says a lot of really smart and interesting things, but what it says that really struck me was that it was suggested using single locus models to describe even complicated non-single locus problems. And I felt especially kind of welcome there because my whole career, since 2016, as long as I've been studying this problem, has been based on a very, very simple single locus analogy that was introduced in 1970 by John Maynard Smith. This notion of a protein space, right? This notion that there's a protein and there's a sequence space. There's a kind of a set of things. There's a set of sequences that this protein can explore. This is really where I've lived since 2016. And to kind of hear now somebody in 2020 say maybe we should be thinking about problems in a single locus context was really, really helpful for me. So to give an example of the type of thing that I do in this, right, we think about kind of something like antibiotic resistance or anti-malarial resistance, more specifically in my case. What you're seeing here is a fitness graph, and we're talking about 4 different mutations constructed in all combinations. So it's a combinatorial complete set of mutations. And we can say this is like a fitness graph, or say it's a fitness landscape or a hypercube or what have you. And really, we construct it this way so we can basically potentially see what direction evolution will evolve. And I've kind of lived, again, with data sets no larger than this. And I think my rationale has always been, I'd rather understand a set of mutations very, 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 very well, right? Whereas a lot of you study these really, really complicated, super nerdy large data sets and are able to say really, really interesting and smart things about them. But I've lived here in large part because this is where the problem that I'm interested in has lived. But thankfully, again, due to Dr. Oman and people like of that nature, maybe this is kind of the scale at which we can understand the larger problem of predicting evolution. Right. So today's lessons, we'll talk about something in particular, how an environment shapes protein space, right, and consequently adaptive trajectories. That is how the environment kind of framing how evolution happens. And more specifically, how epistasis by environment interactions is a critical piece of information for prediction. I'll introduce one key idea, the environmental modulation of epistasis, the epistatic norm of reaction. And we'll come back to that. So evolution through the protein space, we think about this as a way to ask questions about the replay the tape problem. That is evolution repeatable? And repeatability and predictability obviously go hand in hand, right? So the idea is, when you think about something like antibiotic resistance, right, that tends to have this repeatable nature in individual patient over individual patient, we've seen that multiple times. So it's been a kind of a perfect natural experiment to kind of ask basic questions like that. Fortunately, in some diseases, right, it really does take place within a single locus, and so it's really easy to study. We can ask kind of what are the forces that are shaping this? And so we can think about this question more directly. We can look at two different types of environmental gradients, right? We're talking about treating an infectious disease, one enzyme and the sets of mutations. We can talk about drug types. So there's two different types of drugs, right? We call these antifolates because these are kind of interfering with dihydrofolate reductase. And we can talk about drug concentrations of those drug types. And as you see here, these two drugs are quite similar. And so we can ask basic questions. How does variation across drug type and drug concentration shape the way evolution is actually happening, right? Right. And what we see here, and knowing nothing else, and you just know that each of these individual nodes is a different allele, and there's 16 total alleles. Only thing you need to pay attention to here is that at low pure methylamine concentration, you get one solution. At high pure methylamine concentration, you get a different solution. At low cycloguanol concentration, you get one solution. And at high cycloguanol, you get a different solution, meaning evolution is moving differently in different drug environments, right? And of course, they are different, right, in the different drug types. Now, what's provocative about this finding when I first saw it is that these drugs are supposed to be closely related, right? And more specifically, the notion that evolution is doing different things by simply changing drug concentration undermines the notion that evolution is neatly predictable. There's not one solution to the drug resistance problems. There's many contingent upon various specifics of the environment in this context. And so we can kind of ask and dig in further. Okay, well, what is it about the environment? How can we get a better understanding of this problem here? We know different things are happening, but how is it happening? And that's where I turn to the study of epistasis. Okay, now I like the Weinreich definition, Weinreich, Wiley, and Heckendorn land. The surprise at the phenotype when mutations are combined, given the constituent mutations' individual effects. Now we're talking about physiological epistasis. Epistasis has various definitions. We're talking about as in kind of like the mechanistic interaction between mutations in a locus. But like anything, this interaction between mutations can change as a function of environment. Okay, and that's where we're going to focus. And I think that's really where we think a lot of the heavy lifting is going in terms of being able to explain why different things are happening. Let's just look at the fitness of the different alleles. What you're seeing on the left here is one drug, and on the right is another drug. Those are all 16 of the different alleles. Those are whole alleles that we said, and we believe this looks like a norm of reaction, right? On the x-axis there is drug concentration, and on the y-axis there is growth rate, which is like a performance metric. This is a very, very classical norm of reaction from ecology in some ways. And you're basically just saying that the drug, as the drug concentration changes, so do the growth rates. But what if I took this concept of a norm of reaction, right, when we're talking about drugs, and I walked it into epistasis? That I have an epistasis by environment, or I say context summary. Now, what information is this communicating? This is basically talking about the individual mutations and individual interactions between mutations. For example, this 0011 is talking about, right, those last two mutations, the strength of their interaction across the different drug concentrations. And we can do that both for pyrimethamine, one drug, and for another drug. And let's just focus on this one for a second here. What you're seeing is that this third site mutation, what this basically says is that its average effect across genetic backgrounds changes across the drug environments. So it's deleterious at really, really low drug concentrations, but it gets really, 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 really kind of positive. It's a positive effect at higher drug concentrations. And in fact, we would say this is a compensatory mutation. This is a mutation that, right, when you get this mutation at higher drug concentrations, it compensates for the deficits in an enzyme, and it allows it to kind of be able to survive at high drug concentrations. That's really good information. By looking at this mapping, we're now able to kind of resolve better what happens in adaptive evolution. It's this mutation that does a lot of the heavy lifting in high drug concentrations. So really what I've introduced is this notion that protein space meets epistasis meets the norm of reaction. We can think about single locus problems, right? We can compute epistasis across environmental contexts. And really what we've introduced here is an abstraction for tracking how context modulate epistasis. And I'd argue that really is the key to being able to really predict what's going to happen, right, in nature, right? So that if we want to understand what's going to happen in nature, if we're interested in what's going to happen at elevated temperature, or what's going to happen in salinity, or what's going to happen in certain setting and versus others, we really have to dig. We have to understand the way the mutations of interest are interacting with each other. We can't study the problem solely at the whole allele or haplotype level. We have to kind of get a little bit more detailed and mechanistic if we're really going to come up with a good way to understand these problems such that we can predict them. I want to thank a range of collaborators, right? Dan Weinreich in particular, who's had a very big impact on my career, Maggie Epstein, Rafael Guerrero, many others I can't all name. I want to thank the funders and administrative supporters for everything that they've done for supporting this work through the years. I want to thank my new Department of College and Evolutionary Biology at Yale University, my lab, the OG Plexus, the Geek Lab. I want to announce that I'm looking for talented individuals in quantitative and population genetics and ecological genetics, so please contact me if you're interested in mostly postdocs, but even in the post-bac level. I'm kind of flexible. Must be inclusive, collaborative, support, and time. All right, and lastly, I want to thank the Genomic Forecasting Symposium for the invitation to talk. Thank you for your time, and I'll answer any questions that you might have.